Hello, Assalamu alaikum. How are you, everyone? Hope you all are fine. So, hoping that we are going to start our next video, which is about enzyme. In the chapter of enzyme, we will be studying enzyme and the activation energy of enzyme and how it affect the reaction characteristics of the enzymes. And we have mode of action of enzymes, types of enzymes, and then we have enzymes inhibition. First of all, what is an enzyme? Enzyme are the biocatalyst, which means catalyst means that uh, any chemical which speed up the reaction, any chemical which is speeding up a reaction without involving itself into it, without making any chemical change into it, are known as catalyst. And biocatalyst mean the catalysts which are present inside the body, and the biocatalyst present inside the body are commonly known as enzymes. And enzymes are made up of proteins. Energy of activation. In this topic, we'll be discussing that how an enzyme speed up the reaction. It can be any reaction. So let's understand what does any reaction needs to undergo its procedure. In any reaction, we have reactants and final products. So the reactants need a optimum amount of heat to start its reaction, and in the case of reactions which are induced or which are started with the help of enzymes, the activation energy or the heat from the surrounding is not enough for the reactants. So what does an enzyme do? It forms a complex with the reactants and decrease the activation energy required to the reactants. So it actually levels the required activation energy with the activation energy of the surrounding and induce the reaction or starts the reaction. This is how an enzyme work. Enzymes form a complex with the reactants, and I would like to add one more thing, that the covalent bond is broken when the heat is optimum and the level of the activation energy is equalized by enzyme, then covalent bond is usually broken. So if any MCQs you are getting the option that on which bond does enzyme work on, then you can tell them it's the covalent bond. Now we will discuss about the characteristics of enzymes. Before that, I have one point to mention, which is that the enzymes are produced in the protoplasm, which is a portion of our cytoplasm. And cytoplasm, as you know, is the portion of cell, liquid portion inside the cell. Okay, so following are the characteristics of enzymes, in which first characteristics that the enzyme are proteinaceous. As I told you, they are protein in nature, and they may contain a non-protein part as well, but basically they are formed up of proteins, and they react with both acidic and alkaline solution or alkaline reactants. So they don't care if it is acidic or alkaline, they just induce the reaction or start the reaction or speed up the reaction. This is the point number 2. Point number 3 is enzymes can function within a cell or it may function outside the cell. So yeah, enzyme has to function within the cell as well as outside the cell. So the enzymes functioning within the cell are endoenzymes, and enzymes functioning outside the cell are exoenzymes. Now every enzyme is specific for their own reactants. This is the point number 4, specificity. And let me give you an example of lock, as every lock has its specific and its own key, and it cannot be unlocked by any other key. Likewise, every enzymes have their own reactants, and the enzymes are larger in size than the substrate. Number 5 point. Now here substrate is used for the reactants on which enzyme work on. So from now onwards I will use substrate instead of reactants. The site in an enzyme which react with substrate is known as active site. Now the enzymes have a depression in it, in which the substrate bond with it and form the complex which helps speeding up the reaction, and that site, that depression, is known as active site where substrate is attached. Small amount of enzyme can cause reaction to large amount of substrate. So enzyme is required in very small quantity for a large bulk of substrate. There are some ions that help activate the enzyme such as magnesium, chlorine, and substrate concentration and enzyme concentration and pH values can cause inhibition in the activity of enzyme. So there are some factors which inhibit the activity of enzymes. We will discuss them later as well, and they are heat sensitive. Okay, they are heat sensitive, thermolabile. We will study that onwards. They are also pH sensitive. They have no chemical change after the reaction. So I told you in the introduction that the catalysts are those which do not change themselves after the reaction. Let's talk about the mode of action of enzymes. The best explanation given for the mode of action of enzymes is the lock and key theory. In this theory, it is proposed that every enzyme is specific to another substrate, as well as in this working, it tells you that the depression in an enzyme, which is the active site, is very specific to the shape of the substrate. Just like key is specific to a lock and it fits in, if it has the same positioning or same shape, it can work, or if it do not have the same shape, the key is broken or something, it will never work. Here in this example, key is denoted for the substrate and the lock is the enzyme, which as we know, the substrate fits into the enzyme and enzyme is also larger. We have another explanation of the mode of action, which is induced fit model, which also represents that there is a slight minute change in the catalytic reaction of enzymes, and it's not very much noticeable, but it's just slight change which helps the enzyme to become efficient in its catalytic activity, which is speeding up the reaction. Let's talk about the types of enzymes. Enzymes are quite complicated when it comes to its types. First type is the proteoenzymes, the enzymes which are totally made up of proteins. After that, we have holoenzymes, the enzymes which are made up of protein as well as a non-protein part. Both are available. The protein part is known as apoenzyme, is named as apoenzyme, and the non-protein part is known as prosthetic group. P for prosthetic, which is not standing for protein. So this is quite opposite, and that's why it usually confuses us, because prosthetic we feel like it must be for the protein, but it's not for the protein. Now holoenzyme is also divided into two, because the non-protein part can be organic as well as inorganic, so why not we categorize it into two. So the cofactor are the enzymes which are having inorganic prosthetic group. Okay, non-protein group is inorganic, so the enzyme is known as cofactor, or that part of enzyme is known as cofactor. Coenzyme is an enzyme with the organic compound, organic prosthetic group, or it can be just the name of the coenzyme can be used for the enzyme having organic prosthetic group. Let's talk about the factors affecting enzyme. As I mentioned some of the characteristics which we will study later, we will study those in this topic. Concentration of substrate. More the concentration, more the activity of enzyme, but at up to a limit. So after the limit has reached, then the enzyme activity become constant. Okay, increase in the concentration of substrate do not do any work or have no effect on the enzyme. Number two, effect of temperature. Enzymes are very sensitive to temperature. They need optimum temperature in order to work. Higher temperature, in higher temperature the enzymes deactivate, or we can say get destroyed, but the decrease in temperature which is maybe 0 degrees centigrade. If we take an example, its activity is reduced to minimum, but it doesn't destroy. And they are highly active at 37 degrees Celsius. You have to learn that it is highly active at 37 degrees Celsius, and minimum activity at 0 degrees Celsius. And the third option was that at 100 degree or maybe more than 37 degree, the enzymes are going to destroy or deactivate. Effect of pH: optimum pH is required, not more than that, not less than that. Just for example, 1.4 is required for pepsin, which is an enzyme of stomach. So that's how pH effect it is very sensitive to the pH. If it increases or decreases from 1.4, then the enzyme has no activity. Enzymes, activators, and inhibitors. As I told you, we will be studying activators and inhibitors. What are the activators? The organic compound like magnesium activates the phosphate, zinc activates the anhydrase, which are the enzymes. Okay, so these are the activators. Zinc and magnesium compound like this activates the enzymes like phosphate and anhydrase. Inhibitors: enzyme can be inhibit. There are two methods of the inhibition of enzymes, which is competitive, non-competitive, and the third method is feedback inhibition. So first of all, competitive inhibition, the type of inhibition in which there is a competition in between the substrate and the inhibiting substance. This type of inhibiting substance fit into the active site and stops the activity of the enzyme. Non-competitive inhibitor, the type of inhibitor which is non-competitive, which means there is no competition between the substrate and the inhibitor substance. What happens here, that the active site is free and substrate can come to it, attach to it, but the inhibitor substance is attached to any other site rather than the active site. So except leave it, just ignore the active site and it acts on any other site of the enzyme, and the site where the non-competitive inhibitor substance attach is known as allosteric site. So add another word in your dictionary, which is allosteric site. So feedback inhibition, just like you are feeding your brain right now. So feedback inhibition is the inhibition of enzyme due to any feedback, just like for example, just for making your concept, now in the stomach, we got 2 grams of protein in the stomach, and the pepsin was released in a limited quantity, which means for around 100 gram. After it, as we know, the protein is going to finish soon, so 2 grams of protein finished in seconds. Now what to do? Now the feedback is generated to the brain, and then the inhibition is induced to the enzymes, and the pepsin which are extra are inhibited and suppressed. Effect of water: water is necessary for enzyme activity. Without water there is zero enzyme activity. Radiation can alter the shape of the protein. So enzyme, as it is made up of protein, so enzyme can be destructured due to radiations, and which are ultraviolet waves. So here our lecture of enzymes finished, and I would like all of you to subscribe my channel, and I need to reach 1k subscribers as fast as possible. Like the video if it was helpful, and comment below if you have any suggestion. Thank you for watching. Allah Hafiz.